Hello and welcome to Live at PALS. Today we will go over an AS level CIE paper, specifically the May-June 2020 paper 2121. So if you find these videos helpful in preparing, helping you to prepare for your A-level examinations or AS level examinations, please subscribe to our YouTube channel. And let's dive right in and see what the first question holds for us. Okay, so first we start with a figure. Figure 1.1 shows 5 biological molecules. We see a disaccharide here. We have a fatty acid, dipeptide, part of a polysaccharide, and a kind of phospholipid or something here. First question related to these 5 molecules. State the name of the bond in molecule A indicated by the arrow. Okay, so we have a bond between 2 monosaccharides here, and these bonds are called glycosidic bonds. So here we have a glycosidic bond. Glycosidic bonds form when a saccharide bonds to another molecule that can be another saccharide, another carbohydrate, or any other molecule that has an OH group, which would be an O-glycosidic bond that could as well be an NH2 group, for example, that forms the bond. Then we would get an N-glycosidic bond, like in the nucleotides of the DNA. So important is that for glycosidic bonds we have a saccharide that binds to something else. Okay, molecule B is described as a saturated fatty acid. State why molecule B is described as a saturated fatty acid. So here we have the saturated fatty acid, molecule B. What does saturated mean? Saturated means that in our CH structure in the carbon chain, we have only single bonds and no double bonds in the hydrocarbon chain. And each carbon has the maximum number of hydrogen atoms attached to it. The opposite of an unsaturated fatty acid, we can see that here. Molecule E has an unsaturated fatty acid tail. We have the C double bond in this location, in contrary to B, or the second part up here of the second fatty acid tail of molecule E. Because there are no carbon-carbon double bonds in the hydrocarbon chain. Molecule D is a polymer. State the name of the monomer that is used to synthesize this polymer. Okay, that's a bit tricky. What we have to look at here, we can see that the hydrogens at this glycosidic linkage here are pointing upwards, whereas on the second they are pointing downwards, and here they're upwards again, same with the CH2 group here, up, down, up. So this means we have better glucose in this case. Next question, state the letter of the molecule that could be formed during the hydrolysis of a polypeptide. That is C. Here we have a dipeptide, a polypeptide, a protein is hydrolyzed, broken down, decomposed. That is often done by splitting it up into smaller dipeptides. And here we see such a dipeptide, so that is C. State the letter of the molecule that forms part of the cell surface membrane of eukaryotic cells. What are cell membranes mainly made of? They are made of phospholipids, and we see one of these phospholipids. Here we have two fatty acid tails and the phosphate head, which is hydrophilic, the two fatty acid tails hydrophobic, and this allows the formation of the cell membrane, the bilayer of phospholipids, with the hydrophilic phosphate head pointing outwards and the fatty acid tails pointing inwards. E. Okay, part F of the question. That's the last part of question 1. Molecule A and molecule C dissolve in water. Molecule B does not dissolve in water. Explain why molecule A and molecule C dissolve in water, but molecule B does not dissolve in water. That links more or less to what I've already said in the last question, to molecules having parts or groups which are polar, which means they are hydrophilic. They will interact with water to form hydrogen bonds, which causes them to dissolve, and other molecules being hydrophobic, not being able to interact with water because the OH groups are missing, and that leading to the molecule being insoluble in water. So let's see which molecules we're talking about here. A and C dissolve in water, B does not, so A and C, we have these two, they do dissolve in water, whereas B does not dissolve in water. The reason for that is A and B, they have several OH groups, at least a couple of them, that allow, or NH2 here as well, which allows the formation of hydrogen bonds, so these molecules are hydrophilic. Whereas in molecule B we do have an OH group, but this makes up only a tiny part of a very large molecule. This whole part in the back here, the hydrocarbon chain, is hydrophobic, can't form any hydrogen bonds, so this is why molecule B is insoluble in water. A and B have OH groups which are polar, causing the to be hydrophilic. Molecule B has only one OH group and a long non-polar chain which makes the molecule hydrophobic. Okay, let's have a look at question 2. Figure 2 shows some stages of the cell cycle in the meristematic tissue on the root tip of a plant. 3 of these stages, P, Q, and R, are identified in table 2.1. Complete table 2.1 by stating one feature visible in figure 2.1 that is used to identify each stage. So, cell P is in prophase of mitosis. PPP up here. Okay, why is this cell in prophase? Prophase is the first stage of mitosis. This is the stage when the chromosomes start to condense and the nucleus starts to dissolve. Why is cell P in the prophase stage? The reason is that the chromosomes become visible here, but we cannot yet see the distinct legs of the chromosomes. We are not able to see the individual chromatids yet. So we are basically in the process of the condensation and coiling of the DNA to form the chromosomes. Chromosomes start to condense but are not yet visible as an individual fully condensed to chromatid chromosomes. 2 chromatid chromosomes are obviously the chromosomes that have 2 chromatids. So after the synthesis phase, our typical X shaped with the 2 legs and the 2 chromatids being held together in the center at the centromere. Metaphase of mitosis, cell Q. So why is cell Q in metaphase? Metaphase is the second phase of mitosis. This is when the mitotic spindle has transported or moved the chromosomes with the help of the spindle fibers to the equator, or the mitotic plate, or the equatorial plane of the cell. All the chromosomes are lined up here on the equator. Okay, and R is interphase. Interphase is anything that is not mitosis, basically. Interphase is the phase of the cell cycle where the cell spends most time in. It's composed of the G1 S phase and the G2 phase. These are the three phases in between mitosis and cytokinesis, in between the nuclear division and cell division. So why is cell R in the interphase? The reason is there are no chromosomes are visible and the nucleus is present as well. Precise. We can see the nucleolus as well. The nucleolus, the spot that is a bit darker here in the center of the cell, the nucleolus here. So no chromosomes are visible, and the nucleus with the nucleolus inside is present. Is in the form of uncondensed chromatin. Okay, that is more than enough detail to complete the table. What more do we have here? Okay, let's finish that page in the screencast and continue with question 3 and 4 in the next presentation. Draw a labeled diagram to show one chromosome at metaphase of mitosis. On the chromosome you have drawn add labels to show the position of a telomere, a chromatid, and the centromere. Okay, let's draw a chromosome. During the metaphase, this. So where is the telomere? The telomere is at the end of the chromatids. This end bit here, this is a telomere. The complete half of the 2 chromatid chromosome is a chromatid, and at the center here we have the centromere. Telomere, chromatid, and centromere. State the type of protein that is associated with DNA in chromosomes, the proteins around which the DNA is coiled up. are the so-called histones. Describe how the spindle is involved during the process of mitosis. Okay. Describe how the spindle is involved during the process of mitosis. Okay, that question is a bit tougher. So what does the spindle do? So we can start in prophase. During prophase, the spindle fibers attach to the centromeres of the chromosomes. The are moved and arranged at the equator by the spindle. That is then metaphase. During anaphase, the spindle separates the chromosomes at the centromere into sister chromatetes and pulls the teats to the opposite poles of the cell. By decreasing the length of the microtubules attached to the chromatids. Now we could as well write a bit more that during telophase our cell elongates as well. This is achieved by stretching microtubules from one spindle to the other, from one pole to the other, and these microtubules get even further elongated, stretching the cell before the cell finally divides during the last phase of the cell cycle, cytokinesis. Okay, that were the first two questions of paper 21, May-June 2020. I hope I could help you in understanding these two topics, that the questions were related to biological molecules and mitosis and cell division a bit more clearly. I hope you could take something home for your preparation during the screencast. And if this was helpful, please subscribe to our YouTube channel, and I see you for the coming questions in the next screencast. Have a good day.